Hello, I am Dr. Owen On, and welcome to the 5th installment of the boring lecture series. We have transitioned from the musculoskeletal system of the previous session, and we now find ourselves standing at the precipice of the endocrine system, specifically the equine. We will be referencing the 2023 work by Stefaniuk-Szmukier and colleagues regarding the multifactorial genetic influences of EMS, published in Genes, Basel, Volume 14, Issue 8, Article 1544. Equine Metabolic Syndrome, hereafter referred to as EMS, is a clinical and pathophysiological condition of horses and ponies that has gradually emerged as a recognized syndrome over the past several decades, largely in response to increased awareness of obesity, insulin dysregulation, and laminitis in domesticated equine populations. As described by Stefaniuk-Szmukier, Piórkowska, and Ropka-Molik, 2023, EMS is not a single disease entity with a singular cause, but rather a constellation of interrelated metabolic abnormalities that collectively predispose affected animals to clinically significant outcomes, most notably endocrinopathic laminitis. The concept of a syndrome, rather than a discrete disease, is critical to understanding EMS. A syndrome, by definition, refers to a group of signs, symptoms, or pathological features that tend to occur together, without necessarily implying a single, unified etiological pathway. In the case of EMS, this distinction is particularly important, as the syndrome encompasses a wide range of metabolic, endocrine, phenotypic, and genetic variables that interact in complex and sometimes poorly understood ways. We shall now explore the historical context and nomenclatural evolution. The recognition of EMS as a distinct clinical syndrome is relatively recent when compared to many other equine diseases. Historically, horses that would now be classified as having EMS were often described using less specific terminology, such as easy keepers, over-conditioned horses, or animals prone to recurrent laminitis of unclear origin. These descriptive labels, while practically useful, did not reflect an underlying understanding of metabolic dysfunction. The term equine metabolic syndrome was first introduced in 2002 by Johnson, who proposed the designation in part because of its conceptual similarity to human metabolic syndrome. Human metabolic syndrome represents a cluster of risk factors primarily associated with coronary heart disease and type 2 diabetes mellitus. The adoption of parallel terminology in veterinary medicine reflected an emerging recognition that similar metabolic derangements could occur across mammalian species, though with species-specific manifestations and consequences. Prior to the widespread acceptance of the term EMS, various alternative designations were employed in the veterinary literature. These included, but were not limited to, prelaminitic metabolic syndrome, Syndrome X, insulin-resistant syndrome, obesity-associated insulin resistance, and prediabetes mellitus. Each of these terms emphasized different aspects of the condition or reflected different theoretical frameworks for understanding the underlying pathophysiology. However, the proliferation of terminology also created confusion and impeded systematic research. The eventual consensus around the term equine metabolic syndrome provided a unified framework for clinical practice and scientific investigation. In the late 20th and early 21st centuries, advances in endocrinology and veterinary diagnostics began to reveal consistent patterns among these animals. Researchers and clinicians observed that certain horses, particularly ponies and native breeds, exhibited persistent hyperinsulinemia, abnormal responses to oral carbohydrate challenges, and an increased susceptibility to laminitis even in the absence of systemic inflammatory disease or gastrointestinal disturbance. It was within this context that EMS began to be formally conceptualized as a syndrome characterized by insulin dysregulation, obesity or regional adiposity, and laminitis risk. The review by Stefaniuk-Szmukier et al. situates EMS firmly within this modern framework, emphasizing its multifactorial nature and the growing body of evidence implicating genetic predisposition. Let's look at the defining triad of EMS. According to the framework summarized in the Genes Review, EMS is defined by three primary features. Insulin dysregulation, most commonly manifesting as hyperinsulinemia. Increased adiposity, either generalized obesity or regional fat accumulation. A predisposition to laminitis, particularly endocrinopathic laminitis. While these three features are frequently cited together, it is important to note that not all affected animals display each component to the same degree. Some horses may exhibit pronounced insulin dysregulation with minimal visible obesity, whereas others may be markedly obese with more subtle endocrine abnormalities. This variability contributes to the diagnostic challenges associated with EMS and reinforces the concept of the syndrome as a spectrum rather than a binary condition. The authors emphasize that insulin dysregulation is generally regarded as the central pathophysiological abnormality within EMS. Obesity and laminitis, while clinically important, are considered secondary or downstream manifestations that may vary depending on individual susceptibility, management practices, and genetic background. It should be noted that the European College of Equine Internal Medicine, abbreviated as ECEIM, issued a consensus statement in 2019 regarding EMS, which clarified several aspects of the syndrome's definition and diagnostic criteria. This consensus statement, authored by Durham and colleagues, represents the current authoritative reference for clinical practice in Europe and much of the world. The ECEIM statement emphasizes that obesity, while commonly associated with EMS, is not a required feature for diagnosis, and that insulin dysregulation represents the sine qua non of the syndrome. Insulin dysregulation, abbreviated as ID, is a broad term encompassing several abnormalities of insulin secretion, clearance, and tissue responsiveness. In the context of EMS, insulin dysregulation most commonly presents as persistent or exaggerated hyperinsulinemia, particularly in response to dietary carbohydrates. It is important to distinguish insulin dysregulation from insulin resistance, although the two are closely related and often conflated in both clinical practice and scientific literature. Insulin resistance refers specifically to a reduced responsiveness of peripheral tissues, such as skeletal muscle and adipose tissue, to circulating insulin. When tissues are insulin resistant, normal concentrations of insulin are insufficient to elicit appropriate glucose uptake and utilization. Hyperinsulinemia, by contrast, refers to elevated insulin concentrations in the blood, which may occur as a compensatory response to insulin resistance or as a primary abnormality of pancreatic beta cell function. The review highlights that EMS-affected horses may demonstrate insulin dysregulation even in the absence of classic insulin resistance, suggesting that additional mechanisms, including altered incretin signaling and gastrointestinal glucose absorption, may play significant roles. This conceptual distinction represents an important evolution in the understanding of EMS pathophysiology and has implications for both diagnostic testing and therapeutic intervention. Obesity has long been associated with EMS, although the relationship between body condition and metabolic dysfunction is neither linear nor uniform. The paper carefully distinguishes between generalized obesity, assessed using body condition scoring systems, and regional adiposity, which refers to localized fat deposits in specific anatomical regions. Common sites of regional adiposity in EMS-affected horses include the neck, often referred to as a cresty neck, the tail head, the supraorbital fossae, and the sheath or mammary region. These fat deposits are not merely cosmetic features. Rather, they are increasingly recognized as metabolically active tissues capable of secreting adipokines, inflammatory mediators, and other bioactive molecules. The cresty neck, in particular, has received substantial attention in the EMS literature. The Cresty Neck score, developed as a complementary assessment to generalized body condition scoring, provides a semi-quantitative measure of nuchal adiposity on a scale typically ranging from 0 to 5. Research has demonstrated that Cresty Neck scores correlate with metabolic dysfunction independent of overall body condition, suggesting that regional fat distribution may be more metabolically relevant than total adiposity in some individuals. The authors note that some horses with EMS may not appear overtly obese, particularly when assessed using traditional body condition scores. The Hennig body condition scoring system, which employs a 9-point scale with scores of 7 or higher typically considered indicative of obesity, may fail to identify horses with predominantly regional adiposity. This observation underscores the importance of regional fat assessment and supports the notion that EMS cannot be diagnosed solely on the basis of visual appearance. We now turn to laminitis, the primary clinical concern. Laminitis is the most clinically significant and welfare-relevant outcome associated with EMS. It is a painful, debilitating condition affecting the laminar structures of the equine hoof, often resulting in chronic lameness or euthanasia in severe cases. In EMS, laminitis is classified as endocrinopathic, meaning it arises from hormonal or metabolic disturbances rather than systemic inflammation or mechanical overload. The review emphasizes that hyperinsulinemia is strongly implicated in the pathogenesis of endocrinopathic laminitis, although the precise cellular and molecular mechanisms remain incompletely understood. Importantly, not all horses with EMS will develop laminitis, and not all cases of laminitis are attributable to EMS. Nevertheless, the strong association between insulin dysregulation and laminitis risk has made EMS a focal point of laminitis prevention strategies in modern equine practice. Epidemiological studies have demonstrated that ponies are approximately twice as likely to develop pasture-associated laminitis compared to horses, a finding that aligns with the increased prevalence of EMS-associated traits in pony populations. One of the central themes of the Stefaniuk-Szmukier et al. Paper is the multifactorial nature of EMS. The authors stress that EMS arises from the interaction of genetic predisposition, environmental factors, dietary composition, management practices, and individual physiological variability. Environmental contributors include access to energy-dense forage, limited physical activity, and seasonal variations in pasture carbohydrate content. Pasture grasses undergo diurnal and seasonal fluctuations in non-structural carbohydrate content, with concentrations typically highest during periods of active growth in spring and early summer, and during times of environmental stress such as frost or drought. These variations in forage composition can substantially influence metabolic stress in predisposed animals. Management factors such as housing, exercise routines, and feeding strategies further modulate metabolic risk. Horses maintained in stalls with limited opportunity for movement may be at increased risk compared to horses with access to large paddocks or pastures that encourage continuous low-intensity exercise. Similarly, feeding practices that provide concentrated energy sources, such as grain-based concentrates or high-quality hay, may exacerbate metabolic dysfunction in susceptible individuals. Within this complex network of influences, genetic factors are increasingly recognized as playing a substantial role. Certain breeds, particularly ponies and native or primitive horse breeds, exhibit a higher prevalence of EMS-associated traits, suggesting heritable components that influence metabolic efficiency and insulin regulation. We shall now look at the evolutionary perspective. The review briefly alludes to the idea that EMS may represent a maladaptive expression of traits that were once evolutionarily advantageous. Horses evolved as grazing animals adapted to environments characterized by fluctuating food availability and periods of nutritional scarcity. In such environments, efficient energy storage and heightened insulin sensitivity would have conferred survival advantages, allowing animals to accumulate energy reserves during periods of abundance and mobilize these reserves during periods of scarcity. In modern domesticated settings, however, continuous access to high-energy diets and limited physical exertion may transform these adaptive traits into liabilities. The result is a metabolic phenotype that predisposes animals to obesity, insulin dysregulation, and laminitis. This conceptual framework, sometimes referred to as the thrifty genotype hypothesis by analogy to human metabolic disease, provides a useful evolutionary lens through which to understand breed predispositions and individual variability within the EMS spectrum. This conceptual framing does not imply inevitability, but it does provide a useful lens through which to understand breed predispositions and individual variability within the EMS spectrum. Breeds that evolved in harsh environments with limited forage availability, such as Welsh ponies, Shetland ponies, and Icelandic horses, may carry a greater burden of thrifty genotype alleles that become problematic under conditions of nutritional abundance. Conversely, breeds developed in more temperate environments with consistent forage availability, or breeds selected primarily for athletic performance rather than survival under harsh conditions, may carry fewer risk alleles and demonstrate lower EMS prevalence. We now transition to insulin dysregulation and endocrine mechanisms. Insulin dysregulation occupies a central and organizing role in the conceptual framework of equine metabolic syndrome, and for this reason it warrants prolonged, careful, and highly detailed examination. In the review by Stefaniuk-Szmukier, Piórkowska, and Ropka-Molik, 2023, insulin dysregulation is repeatedly emphasized as the most consistent and defining physiological abnormality observed in horses diagnosed with EMS. While obesity and laminitis are clinically conspicuous, insulin dysregulation is the underlying metabolic feature that links these manifestations into a coherent syndrome. Insulin is a peptide hormone synthesized and secreted by the beta cells of the pancreatic islets of Langerhans. In horses, as in other mammals, insulin plays a fundamental role in maintaining glucose homeostasis by facilitating the uptake of glucose into insulin-sensitive tissues, particularly skeletal muscle and adipose tissue, while simultaneously suppressing hepatic glucose production through gluconeogenesis and glycogenolysis. The insulin molecule itself consists of two polypeptide chains, designated A and B, connected by disulfide bonds. Insulin is initially synthesized as a single-chain precursor molecule called pre-proinsulin, which is subsequently processed to proinsulin within the endoplasmic reticulum, and finally cleaved to produce mature insulin and C-peptide. The measurement of C-peptide can provide information about endogenous insulin secretion, as it is secreted in equimolar amounts with insulin but has a longer half-life and is not cleared by the liver during first-pass metabolism. Under normal physiological conditions, insulin secretion is tightly regulated by circulating glucose concentrations through a sophisticated sensing mechanism within pancreatic beta cells. Following ingestion of carbohydrates, blood glucose levels rise, triggering glucose uptake into beta cells via GLUT2 glucose transporters. Intracellular glucose metabolism leads to increased ATP production, closure of ATP-sensitive potassium channels, membrane depolarization, calcium influx, and ultimately insulin vesicle exocytosis. This insulin then promotes glucose uptake in peripheral tissues, leading to a subsequent decline in blood glucose levels and a corresponding reduction in insulin secretion, thereby completing a negative feedback loop. Insulin exerts its biological effects through binding to specific insulin receptors, abbreviated IRS, on the surface of target cells. These receptors are transmembrane receptor tyrosine kinases that, upon insulin binding, undergo autophosphorylation and initiate a complex downstream signaling cascade. Key signaling pathways include the phosphatidylin 3 kinase pathway, abbreviated PI3K, which mediates metabolic effects such as glucose transport and glycogen synthesis, and the mitogen-activated protein kinase pathway, which mediates growth and proliferative responses. In EMS, this finely tuned regulatory system becomes disrupted. The review emphasizes that horses with EMS frequently exhibit abnormal insulin responses that are disproportionate to glucose concentrations, particularly following oral carbohydrate intake. Insulin dysregulation is not a singular pathological process, but rather an umbrella term encompassing multiple abnormalities related to insulin metabolism. In the context of EMS, insulin dysregulation may include basal hyperinsulinemia, defined as elevated fasting insulin concentrations, exaggerated insulin responses to oral or intravenous glucose challenges, prolonged duration of postprandial hyperinsulinemia, altered insulin clearance, primarily hepatic, impaired tissue responsiveness to insulin, traditionally termed insulin resistance, altered incretin hormone secretion or action. The authors highlight that insulin dysregulation may exist independently of overt insulin resistance, challenging earlier assumptions that insulin resistance was a necessary prerequisite for EMS. This distinction is subtle but important, as it broadens the scope of potential mechanisms contributing to hyperinsulinemia and has implications for diagnostic testing strategies. Hyperinsulinemia, defined as persistently elevated circulating insulin concentrations, is widely regarded as the hallmark endocrine abnormality in EMS. Horses with EMS may exhibit elevated fasting insulin levels, although many display normal fasting values but exaggerated postprandial insulin responses. This heterogeneity in presentation has led to the development of dynamic testing protocols designed to unmask insulin dysregulation that may not be apparent under basal conditions. The review discusses how hyperinsulinemia can arise through several pathways. One mechanism involves compensatory insulin secretion in response to reduced peripheral insulin sensitivity. When tissues become resistant to insulin's effects, pancreatic beta cells increase insulin output to maintain glucose homeostasis. This compensatory hyperinsulinemia may be sufficient to maintain normoglycemia for extended periods, masking the underlying metabolic dysfunction until beta cell exhaustion occurs or other precipitating factors intervene. Another mechanism involves dysregulated insulin secretion driven by gastrointestinal factors, independent of peripheral glucose uptake. This mechanism, discussed in greater detail below, implicates the incretin hormone system and suggests that some forms of hyperinsulinemia may arise from excessive enteric stimulation of insulin secretion rather than from peripheral tissue resistance. Importantly, experimental studies have demonstrated that sustained hyperinsulinemia alone, even in the absence of hyperglycemia, is sufficient to induce laminitis. Delat and colleagues conducted a seminal study in which clinically normal horses were subjected to prolonged euglycemic hyperinsulinemia through intravenous insulin infusion while maintaining normal blood glucose concentrations through concurrent glucose infusion. All experimental horses developed laminitis within approximately 48 hours, with early clinical signs appearing at around 31.5 hours. This finding underscores the pathological significance of insulin itself, rather than glucose, in the development of endocrinopathic laminitis, and represents a paradigm shift in the understanding of laminitis pathogenesis. We shall now explore the evolution from insulin resistance to insulin dysregulation. ION. Historically, insulin resistance was considered the primary metabolic defect underlying EMS. Insulin resistance refers specifically to a reduced responsiveness of insulin-sensitive tissues to the biological effects of insulin. In insulin-resistant horses, normal concentrations of insulin are insufficient to elicit appropriate glucose uptake, prompting compensatory increases in insulin secretion. The concept of insulin resistance was borrowed largely from human metabolic disease, where peripheral tissue insulin resistance plays a central role in the pathophysiology of type 2 diabetes mellitus and metabolic syndrome. Early research in equine metabolism naturally adopted this framework, and much of the initial EMS literature focused on measuring insulin sensitivity using various techniques, including euglycemic hyperinsulinemic clamps, frequently sampled intravenous glucose tolerance tests, and combined glucose-insulin tests. However, the authors note that insulin resistance does not fully account for the metabolic abnormalities observed in EMS. Some horses demonstrate marked hyperinsulinemia despite preserved insulin sensitivity, as measured by gold standard techniques. This observation has led to a conceptual shift away from insulin resistance as the sole explanatory model and toward a broader understanding of insulin dysregulation that includes gastrointestinal and hormonal influences. This evolution in terminology reflects a more nuanced appreciation of the complexity of insulin homeostasis. Rather than attributing all hyperinsulinemia to peripheral tissue resistance, the field now recognizes that multiple mechanisms may contribute, including excessive insulin secretion, reduced insulin clearance, and altered incretin signaling. The term insulin dysregulation accommodates this mechanistic diversity and avoids the implicit assumption that resistance is the primary underlying defect. And now onto the enteroinsular axis and incretin hormones. One of the more detailed and mechanistically rich discussions in the review concerns the role of the gastrointestinal tract in insulin dysregulation. The gastrointestinal tract is not merely a passive conduit for nutrient absorption. It is an active endocrine organ that influences systemic metabolism through the secretion of incretin hormones. Incretins are gastrointestinal peptides released in response to nutrient ingestion. The two primary incretin hormones discussed in the context of EMS are glucose-dependent insulinotropic polypeptide, abbreviated GIP, glucagon-like peptide 1, abbreviated GLP-1. These hormones potentiate insulin secretion from pancreatic beta cells in a glucose-dependent manner, a phenomenon known as the incretin effect. In humans, the incretin effect accounts for approximately 50 to 70% of the insulin response to oral glucose administration, compared to intravenous glucose administration, which bypasses the gastrointestinal tract and does not stimulate incretin release. GIP is a 42 amino acid peptide hormone synthesized and secreted by enteroendocrine K cells located primarily in the duodenum and proximal jejunum. GIP secretion is stimulated by the presence of glucose, fatty acids, and amino acids in the intestinal lumen. Beyond its insulinotropic effects, GIP also stimulates glucagon secretion, promotes fat deposition in adipose tissue, and may influence bone metabolism. The GIP receptor is expressed on pancreatic beta cells, adipocytes, and various other tissues. GLP-1 is a 30 amino acid peptide derived from post-translational processing of the proglucagon gene in intestinal L cells, which are located primarily in the ileum and colon. GLP-1 secretion is stimulated by nutrient ingestion, with carbohydrates, fats, and proteins all serving as secretagogues. In addition to potentiating glucose-dependent insulin secretion, GLP-1 inhibits glucagon secretion, delays gastric emptying, and promotes satiety through central nervous system effects. The GLP-1 receptor is expressed on pancreatic beta cells, gastrointestinal tissues, and various regions of the brain. In horses with EMS, exaggerated incretin responses to oral carbohydrate intake have been observed, leading to disproportionate insulin secretion. Research by Delat and colleagues demonstrated that approximately 23% of the variation in insulin concentrations following oral non-structural carbohydrate administration could be explained by variations in active GLP-1 concentrations. This finding suggests that altered incretin signaling may play a significant role in postprandial hyperinsulinemia, particularly in horses that do not exhibit classic insulin resistance as measured by peripheral tissue glucose uptake. The review suggests that altered incretin signaling may play a significant role in postprandial hyperinsulinemia, particularly in horses that do not exhibit classic insulin resistance. This mechanism provides a plausible explanation for why some EMS-affected horses demonstrate normal insulin sensitivity, as measured by techniques that assess peripheral tissue responsiveness, but abnormal insulin dynamics following feeding. It also explains why oral glucose or sugar challenges may be more sensitive than intravenous glucose tolerance tests for detecting insulin dysregulation in EMS. The distinction between oral and intravenous glucose administration is particularly informative in the study of EMS and has important implications for diagnostic testing. When glucose is administered orally, it stimulates both glucose absorption and incretin release from the gastrointestinal tract. Intravenous glucose administration, by contrast, bypasses the gastrointestinal tract entirely and does not stimulate incretin secretion. Studies referenced in the review have shown that some EMS horses exhibit exaggerated insulin responses to oral glucose challenges, but relatively normal responses to intravenous glucose. This discrepancy supports the hypothesis that gastrointestinal factors contribute significantly to insulin dysregulation in at least a subset of affected animals. From a diagnostic perspective, this distinction has practical implications. Oral sugar tests, which involve administration of corn syrup or other simple sugars followed by serial insulin measurements, may reveal abnormalities not detected by intravenous testing methods such as the frequently sampled intravenous glucose tolerance test. The ECEIM consensus statement recommends oral testing as a practical and sensitive method for detecting insulin dysregulation in clinical practice, acknowledging the logistical advantages and physiological relevance of testing under conditions that mimic normal feeding. The oral sugar test typically involves administration of 0.15 milliliters of corn syrup per kilogram body weight, or approximately 15 milliliters of corn syrup per 100 kilograms body weight, via oral syringe. Blood samples are collected before and at 60 and 90 minutes after corn syrup administration. Insulin concentrations exceeding 60 microinternational units per milliliter at 60 or 90 minutes are generally considered indicative of insulin dysregulation, although specific cutoff values may vary depending on assay methodology and population characteristics. In addition to increased insulin secretion, altered insulin clearance may contribute to hyperinsulinemia in EMS. Insulin clearance occurs primarily in the liver, where approximately 50% of circulating insulin is removed from the bloodstream during first-pass metabolism. Insulin is internalized via receptor-mediated endocytosis and subsequently degraded by intracellular proteases, particularly insulin-degrading enzyme. The review briefly notes that reduced hepatic insulin clearance could theoretically contribute to elevated circulating insulin concentrations. While direct evidence in horses remains limited, this mechanism has been documented in other species and represents an area of ongoing research interest. In humans with type 2 diabetes and metabolic syndrome, reduced hepatic insulin clearance has been identified as a contributing factor to hyperinsulinemia, particularly in early stages of disease when beta cell secretory capacity remains intact. Factors that may influence hepatic insulin clearance in horses include hepatic steatosis, or fatty infiltration of the liver, which is increasingly recognized as a component of EMS pathophysiology. Adipose tissue dysfunction and increased circulating fatty acids may promote hepatic fat accumulation, which in turn may impair insulin receptor function and insulin degradation in hepatocytes. However, The relative contribution of reduced insulin clearance to hyperinsulinemia in EMS remains to be fully elucidated and represents an area requiring further investigation. Chronic hyperinsulinemia exerts widespread effects on multiple tissues beyond its immediate metabolic consequences. In skeletal muscle, prolonged exposure to high insulin concentrations may lead to downregulation of insulin receptors or impairment of post-receptor signaling pathways, a phenomenon known as insulin-induced insulin resistance. This creates a vicious cycle wherein hyperinsulinemia both results from and contributes to insulin resistance, further exacerbating metabolic dysregulation. In adipose tissue, hyperinsulinemia promotes lipogenesis and inhibits lipolysis, contributing to fat accumulation. Insulin stimulates fatty acid synthase, the enzyme responsible for de novo lipid synthesis, and inhibits hormone-sensitive lipase, the enzyme responsible for triglyceride breakdown. This effect reinforces the association between insulin dysregulation and obesity, although the temporal relationship between these phenomena remains complex and bidirectional. Some horses may develop hyperinsulinemia secondary to obesity, while others may develop obesity secondary to hyperinsulinemia, and in many cases, both processes likely operate simultaneously. In the liver, chronic hyperinsulinemia may promote hepatic steatosis through increased de novo lipogenesis and impaired fatty acid oxidation. Insulin stimulates the transcription factor sterol regulatory element binding protein 1C, abbreviated SREB1C, which upregulates genes involved in fatty acid synthesis. Simultaneously, hyperinsulinemia may suppress the expression of genes involved in fatty acid oxidation, leading to net lipid accumulation in hepatocytes. The authors emphasize that hyperinsulinemia should not be viewed solely as a compensatory response, but rather as an active driver of pathological processes within EMS. This perspective has important therapeutic implications, as interventions aimed at reducing insulin concentrations may have benefits beyond simply addressing the underlying insulin resistance or dysregulation. Seasonal variation in insulin sensitivity and secretion is briefly addressed in the review and represents an important consideration in EMS management. Horses often exhibit reduced insulin sensitivity during periods of abundant pasture growth, particularly in spring and early summer when non-structural carbohydrate content of forage is high. This seasonal pattern reflects the natural fluctuation in pasture carbohydrate concentrations, which are influenced by environmental factors including temperature, light exposure, moisture availability, and stage of plant growth. During periods of active growth under favorable conditions, grasses accumulate simple sugars and fructans, which serve as energy storage compounds. These non-structural carbohydrates are highly digestible and can trigger substantial postprandial insulin responses. This seasonal pattern interacts with genetic predisposition and management practices to influence the severity and expression of insulin dysregulation. Horses with EMS may experience pronounced worsening of insulin dynamics during these periods, increasing their risk of laminitis. Clinical experience and epidemiological studies have demonstrated that laminitis associated with EMS often occurs in spring and early summer, coinciding with peak pasture carbohydrate availability. Additionally, some research suggests that horses may exhibit photoperiod-related changes in insulin sensitivity independent of dietary carbohydrate intake. Longer day length may be associated with reduced insulin sensitivity through mechanisms that remain incompletely understood but may involve seasonal changes in activity levels, energy balance, or neuroendocrine regulation. In summary, insulin dysregulation in EMS is a multifaceted phenomenon involving excessive insulin secretion from pancreatic beta cells, altered tissue responsiveness to insulin effects, gastrointestinal incretin effects that amplify insulin responses to oral nutrients, possible reductions in hepatic insulin clearance, tissue-specific adaptations to chronic hyperinsulinemia, seasonal fluctuations in insulin dynamics. The review by Stefaniuk-Szmukier et al. Underscores that no single mechanism fully accounts for insulin dysregulation in EMS. Rather, it arises from the convergence of multiple endocrine and metabolic pathways, modulated by genetic and environmental factors. This complexity necessitates a multifaceted approach to diagnosis, incorporating both basal and dynamic testing, consideration of seasonal influences, and recognition that different individuals may manifest insulin dysregulation through different primary mechanisms. This detailed understanding of insulin dysregulation sets the stage for subsequent discussions of laminitis pathophysiology, adipose tissue function, and genetic susceptibility, each of which builds upon the endocrine abnormalities described here. We shall now transition to laminitis as a pathophysiological and clinical consequence. Laminitis represents the most clinically significant and welfare-compromising outcome associated with equine metabolic syndrome. While insulin dysregulation and adiposity may persist for extended periods with minimal outward clinical signs, laminitis often manifests acutely and dramatically, drawing immediate attention to the underlying metabolic derangements. For this reason, laminitis occupies a central position in both the clinical definition and practical importance of EMS. In the review by Stefaniuk-Szmukier, Piórkowska, and Ropka-Molik, 2023, laminitis is discussed primarily as an endocrinopathic condition arising from metabolic dysfunction rather than from inflammatory or mechanical causes. This distinction is essential, as it frames laminitis not merely as a disease of the hoof, but as a systemic consequence of endocrine imbalance. To understand laminitis in the context of EMS, it is necessary to review, in some detail, the anatomy of the equine hoof. The hoof capsule, composed of keratinized epidermis, encases the distal phalanx, also known as the coffin bone or third phalanx, and is connected to it via an intricate interdigitating network of epidermal and dermal lamellae. These lamellae serve both structural and physiological functions. Structurally, they suspend the distal phalanx within the hoof capsule, distributing the animal's weight across a large surface area and allowing forces to be transmitted from the skeleton to the ground surface without direct bone-to-ground contact. The lamellar interface consists of approximately 600 primary epidermal lamellae, each giving rise to approximately 100 to 200 secondary lamellae, resulting in a total surface area of approximately 0.8 square meters. This enormous surface area provides a secure attachment between the distal phalanx and the hoof wall while allowing for continuous growth and remodeling. Physiologically, the lamellae contain a rich vascular and cellular network that supports continuous tissue remodeling and repair. The basement membrane at the dermal-epidermal interface serves as a critical structural element, anchoring epithelial cells to the underlying dermis through hemidesmosomes and focal adhesions. The integrity of this basement membrane and the health of the lamellar epithelial cells are essential for maintaining normal hoof function. Laminitis refers to pathological changes affecting these lamellar structures. These changes may include cellular dysfunction, inflammation, vascular dysregulation, basement membrane degradation, and separation of lamellar attachments. In severe cases, complete failure of the suspensory apparatus can occur, leading to displacement or rotation of the distal phalanx within the hoof capsule, a condition associated with severe pain and poor prognosis. We shall now discuss classification and terminology of laminitis. Laminitis can be broadly classified into several categories based on underlying etiology. Traditional classifications include sepsis-associated laminitis, often following gastrointestinal disease, particularly large intestinal carbohydrate overload or colitis. Supporting limb laminitis, resulting from prolonged weight bearing on a single limb due to contralateral limb injury. Endocrinopathic laminitis, associated with metabolic and hormonal disorders, including EMS and pituitary pars intermedia dysfunction, abbreviated PPID. Traumatic laminitis, resulting from mechanical trauma such as excessive concussion on hard surfaces. EMS-related laminitis falls into the endocrinopathic category, alongside laminitis associated with PPID, formerly known as equine Cushing's disease. The review emphasizes that endocrinopathic laminitis differs mechanistically from inflammatory forms of laminitis, despite sharing similar clinical signs. This mechanistic distinction has important implications for both prevention and treatment strategies. Laminitis can also be classified temporally, with distinctions made between acute, subacute, and chronic phases. The acute phase is characterized by the initial onset of clinical signs, typically including lameness, reluctance to move, characteristic weight shifting or laminitic stance, increased digital pulses, and increased hoof wall temperature. The subacute phase refers to ongoing disease that may wax and wane in severity, but has not resulted in permanent structural changes. Chronic laminitis is characterized by persistent or recurrent clinical signs associated with permanent structural changes within the hoof, including distal phalanx displacement, rotation, or sinking. Clinically, laminitis associated with EMS may present acutely or chronically. Acute episodes are characterized by sudden onset of lameness, reluctance to move, characteristic posturing with weight shifted toward the heels to relieve pressure on the toe region, and increased digital pulses. Physical examination may reveal increased hoof wall temperature, pain in response to hoof testers applied to the sole, and a stiff, stilted gait. In severe cases, horses may be reluctant or unable to stand, and may spend prolonged periods in recumbency. Chronic laminitis may manifest as recurrent, low-grade lameness, progressive hoof capsule distortion, including divergent growth rings that are wider at the heel than at the toe, flattening or convexity of the sole, and radiographic evidence of distal phalanx displacement. The hallmark radiographic finding in chronic laminitis is rotation of the distal phalanx relative to the hoof wall, measured as the angle between the dorsal hoof wall and the dorsal surface of the distal phalanx. In normal horses, these two structures should be parallel or nearly parallel. One notable feature of EMS-associated laminitis is that it may develop in horses that otherwise appear systemically healthy. Unlike sepsis-associated laminitis, there may be no preceding illness, fever, diarrhea, colic, or systemic inflammatory response. The horse may be bright, alert, and maintaining normal appetite and attitude at the time laminitis develops. This insidious nature complicates early detection and underscores the importance of identifying metabolic risk factors before clinical laminitis develops. Additionally, many horses with EMS experience mild, subclinical laminitis episodes before severe clinical disease is recognized. These subclinical episodes may cause only subtle gait changes, slight reluctance to move on hard surfaces, or mild sensitivity to hoof testers, signs that may be overlooked or attributed to other causes. Over time, however, repeated subclinical episodes can result in cumulative structural damage, eventual distal phalanx displacement, and overt clinical disease. Recognition of this subclinical disease phase has led to increased emphasis on proactive metabolic screening in at-risk populations. We shall now discuss hyperinsulinemia as the primary driver of endocrinopathic laminitis. The review places particular emphasis on experimental and clinical evidence linking hyperinsulinemia to laminitis. As mentioned previously, studies in which horses were subjected to prolonged, experimentally induced hyperinsulinemia have demonstrated the development of laminitis in the absence of hyperglycemia or systemic inflammation. The landmark study by de Laat and colleagues, published in 2010 in Equine Veterinary Journal, subjected healthy standardbred horses to 48 hours of euglycemic hyperinsulinemia through continuous intravenous insulin and glucose infusion. All experimental horses developed histological and clinical evidence of laminitis. Early clinical symptoms appeared after a median of 31.5 hours and included significantly increased heart rate, anxiety, episodic shifting of weight between feet, stiff gait at the walk, and reluctance to trot. Histological examination of hoof tissues collected after the experimental period revealed lamellar epithelial cell proliferation, basement membrane disruption, and lamellar stretching and separation. These findings represent a significant shift in the understanding of laminitis pathogenesis. Rather than being solely a consequence of vascular compromise or inflammation, laminitis in EMS appears to be driven, at least in part, by direct effects of insulin on lamellar tissues. This discovery has fundamentally altered the approach to laminitis prevention and has directed research attention toward understanding the cellular and molecular mechanisms by which insulin induces lamellar pathology. Several hypotheses have been proposed to explain how hyperinsulinemia leads to laminar pathology. The review summarizes these mechanisms without asserting a single definitive pathway, reflecting the current state of incomplete understanding in the field. One proposed mechanism involves insulin-like growth factor 1, abbreviated IGF-1, receptor activation. Insulin and IGF-1 are structurally similar peptide hormones with homologous receptors. At high concentrations, insulin may bind to IGF-1 receptors in lamellar tissues, promoting abnormal cellular proliferation and weakening of lamellar attachments. Immunohistochemical studies have demonstrated that while insulin receptors are not abundant on lamellar epithelial cells, IGF-1 receptors are present in substantial numbers. In vitro studies have shown that insulin can stimulate proliferation of cultured lamellar epithelial cells through IGF-1 receptor activation, supporting this hypothesis. A second hypothesis focuses on vascular dysregulation. Insulin has vasoactive properties and influences endothelial nitric oxide synthase activity. Binding of insulin to endothelial insulin receptors activates nitric oxide synthase, leading to increased production of nitric oxide, a potent vasodilator. Chronic hyperinsulinemia may alter blood flow within the laminar microvasculature through dysregulated nitric oxide production. Some researchers have proposed that excessive vasodilation may lead to increased digital perfusion, edema formation within the laminar tissues, and ultimately mechanical failure of the lamellar attachments. Others have suggested that insulin resistance in vascular endothelium may impair normal vasodilatory responses, leading to vasoconstriction, tissue ischemia, and hypoxic injury. A third proposed mechanism involves alterations in glucose transport and cellular metabolism within lamellar epithelial cells. The lamellarly expressed glucose transporter 1, abbreviated GLUT1, which facilitates insulin-independent glucose uptake. Under conditions of altered glucose availability or metabolism, lamellar epithelial cells may experience energetic stress. Disrupted energy metabolism may impair the structural integrity and regenerative capacity of lamellar tissues. This hypothesis is supported by in vitro studies demonstrating that glucose deprivation causes separation of lamellar explants. A fourth mechanism involves the formation of advanced glycation end products, abbreviated AGEs. Even in the absence of overt hyperglycemia, horses with EMS may experience fluctuations in tissue glucose concentrations. Glucose can react non-enzymatically with proteins to form AGEs through a series of reactions known collectively as glycation. Methylglyoxal, a reactive glucose metabolite, is a particularly potent glycating agent. AGEs can accumulate in tissues and cause structural damage through cross-linking of extracellular matrix proteins and activation of inflammatory signaling pathways. Ex vivo studies using equine lamellar explants have demonstrated that methylglyoxal exposure leads to loss of mechanical strength and increased expression of matrix metalloproteinases, enzymes that degrade extracellular matrix components. A fifth mechanism involves matrix metalloproteinase, abbreviated MMP, activation. MMPs are a family of proteolytic enzymes that degrade various components of the extracellular matrix, including collagens, laminins, and fibronectin. Under normal conditions, MMP activity is tightly regulated by tissue inhibitors of metalloproteinases, abbreviated TIMPs. In laminitis, the balance between MMPs and TIMPs becomes disturbed, leading to excessive matrix degradation. Studies have demonstrated increased MMP2 and MMP9 expression in laminitic tissues, and inhibition of MMPs has been shown to reduce lamellar damage in experimental models. The authors note that these mechanisms are not mutually exclusive and may operate simultaneously or sequentially. Current evidence suggests that insulin-induced laminitis likely involves multiple pathophysiological pathways that converge on common endpoints: lamellar epithelial dysfunction, basement membrane degradation, and mechanical failure of the suspensory apparatus. Unraveling the relative contributions of these various mechanisms remains an active area of research. The distinction between endocrinopathic and inflammatory laminitis is repeatedly emphasized in the review and has important clinical implications. Inflammatory laminitis, such as that occurring secondary to sepsis or carbohydrate overload, is characterized by marked inflammatory mediator release, leukocyte infiltration, activation of inflammatory signaling pathways, and systemic signs of illness. By contrast, endocrinopathic laminitis often lacks overt inflammation, particularly in early stages. Histological studies have demonstrated minimal inflammatory cell infiltration in lamellar tissues during insulin-induced laminitis, suggesting a fundamentally different pathological process. While some degree of inflammation may develop secondarily as tissues become damaged, the primary insult appears to be metabolic rather than inflammatory. This distinction has important implications for treatment and prevention. Anti-inflammatory therapies alone, such as non-steroidal anti-inflammatory drugs, may provide symptomatic relief but are insufficient to address the underlying endocrine drivers of EMS-associated laminitis. Effective management requires control of hyperinsulinemia through dietary modification, weight management, and, in some cases, pharmacological intervention. Conversely, aggressive anti-inflammatory therapy, while appropriate for sepsis-associated laminitis, may be less critical in endocrinopathic cases. One of the more challenging aspects of EMS-related laminitis is the temporal disconnect between metabolic dysfunction and clinical disease. Insulin dysregulation and adiposity may be present for months or years before the onset of laminitis. During this period, affected horses may appear clinically normal, with no overt signs of metabolic disease. The review highlights that laminitis often represents a tipping point, triggered by additional factors such as dietary changes, seasonal pasture growth, concurrent illness, or stress. This delayed manifestation complicates causal inference and may lead to underestimation of metabolic risk in apparently healthy animals. Owners may not recognize that their horse has EMS until laminitis develops, at which point significant and potentially irreversible lamellar damage may have already occurred. Not all laminitic episodes in EMS are severe or immediately obvious. Some horses experience recurrent, subclinical laminitis characterized by subtle gait changes, reluctance to move on hard or uneven surfaces, increased sensitivity to hoof testers, or progressive changes in hoof conformation. These subtle signs may not prompt veterinary evaluation, and the horse may continue to be ridden or worked with unrecognized discomfort. Over time, repeated subclinical episodes may result in chronic laminar damage, even in the absence of acute clinical crises. The review suggests that such chronic changes may contribute to long-term morbidity and reduced performance. Recognition of subclinical disease has led to increased emphasis on early metabolic screening, regular hoof evaluation, and proactive management to prevent progression to overt laminitis. Diagnosing laminitis in the context of EMS presents several challenges. Early-stage laminitis may be difficult to detect, particularly in stoic animals or those with mild clinical signs. Physical examination findings may be subtle, and owners may attribute minor gait changes to other causes such as foot soreness from hard ground or minor musculoskeletal injury. Furthermore, laminitis may be the first indication that an animal has underlying metabolic dysfunction. In such cases, metabolic evaluation often occurs only after irreversible laminar damage has already occurred. This diagnostic lag reinforces the importance of proactive metabolic screening in at-risk populations, a theme that recurs throughout the review. Radiographic evaluation can be helpful in documenting the presence and severity of laminitis, but radiographic changes may lag behind clinical and histological changes. Early laminitis may be present without radiographic abnormalities, and radiographs may appear normal even in horses with significant lamellar pathology. Conversely, chronic radiographic changes may persist long after active disease has resolved, complicating assessment of current disease activity. In summary, laminitis serves as both a defining feature and a major clinical consequence of equine metabolic syndrome. While insulin dysregulation represents the central metabolic abnormality, laminitis is the outcome that most directly affects animal welfare and owner concern. The pathophysiology of insulin-induced laminitis involves multiple proposed mechanisms, including IGF-1 receptor activation, vascular dysregulation, altered cellular metabolism, advanced glycation end product formation, and matrix metalloproteinase activation. The review by Stefaniuk-Szmukier et al. Underscores that effective prevention and management of EMS must prioritize the identification and control of laminitis risk through metabolic regulation rather than relying solely on symptomatic treatment of hoof pathology. Early recognition of insulin dysregulation, proactive dietary management, weight control, and regular monitoring of at-risk animals are essential strategies for preventing the devastating consequences of EMS-associated laminitis. We now move on to obesity, adipose tissue, and cellular dysfunction. Obesity has long been recognized as a common, though not universally present, feature of equine metabolic syndrome. In the review by Stefaniuk-Szmukier, Piórkowska, and Ropka-Molik, 2023, obesity is treated not merely as an excess of body mass, but as a metabolically active and biologically influential condition that interacts closely with insulin dysregulation and genetic predisposition. This section examines obesity in EMS with particular attention to adipose tissue distribution, cellular behavior, and molecular consequences. Obesity in horses is typically defined as excessive accumulation of adipose tissue relative to optimal body condition. In clinical practice, obesity is often assessed using body condition scoring systems, most commonly the 9-point Hennig scale, developed originally for horses but applicable to ponies as well. This scale assigns scores from 1, emaciated, to 9, extremely obese, with scores of 4 to 6 generally considered ideal, scores of 7 to 8 indicating overweight to obese condition, and score 9 indicating extreme obesity. The Hennig system evaluates fat deposition at 6 key anatomical locations: along the neck, behind the shoulder, behind the withers, over the ribs, along the back, and at the tail head. Palpation of these areas allows subjective assessment of subcutaneous fat depth and overall body condition. While useful and widely employed, the system has limitations, including subjective interpretation, inter-observer variability, and potential insensitivity to regional fat distribution patterns. However, the review emphasizes that reliance on generalized body condition scores alone may be misleading in the context of EMS. Some horses with EMS may have body condition scores within the moderate range, typically 5 to 6, yet still exhibit significant metabolic dysfunction. This observation has led to increased attention on regional adiposity rather than generalized obesity. Additional assessment methods include morphometric measurements, such as body weight, heart girth circumference, and calculation of body mass index equivalents. Some researchers have proposed weight-to-height ratios as indicators of obesity. However, these methods have not gained widespread clinical adoption and require validation across diverse breed types and body conformations. Regional adiposity refers to the preferential accumulation of fat in specific anatomical locations. In EMS, characteristic sites of fat deposition include the dorsal neck region, commonly referred to as a cresty neck, the tail head and along the croup, the supraorbital fossae, creating prominent fat pads above the eyes, the preputial region in males or mammary region in females, along the back and loin, the pectoral region between the forelimbs. Among these, the cresty neck has received particular attention. The cresty neck score is a semi-quantitative assessment tool that specifically evaluates nuchal adiposity on a scale from 0 to 5. Score 0 indicates no palpable crest, with fat deposited only at the base of the neck. Score 1 indicates a slight crest with minimal fat filling the neck region. Score 2 represents a moderate crest, with fat deposition approximately halfway up the neck. Score 3 indicates a well-developed crest, with fat filling 2/3 or more of the neck region. Score 4 represents a large crest that may fall to one side. Score 5 indicates a very large crest that folds permanently to one side and may hinder flexion of the neck. The review notes that the cresty neck score often correlates with metabolic risk more closely than generalized body condition score. Research by Carter and colleagues demonstrated that cresty neck score was significantly associated with insulin concentrations and hyperinsulinemia, independent of overall body condition score. This finding suggests that nuchal fat may be particularly metabolically active or may serve as a marker for systemic metabolic dysfunction. Regional adiposity is not merely a passive storage of energy. These localized fat depots are metabolically active and may differ in their endocrine and inflammatory profiles compared to subcutaneous fat distributed elsewhere in the body. Adipose tissue function varies by anatomical location, with different depots exhibiting distinct patterns of adipokine secretion, inflammatory cytokine production, lipolytic activity, and insulin sensitivity. A central theme in modern metabolic research, echoed in the review, is the recognition of adipose tissue as an endocrine organ rather than an inert energy storage depot. Adipocytes secrete a variety of biologically active molecules collectively referred to as adipokines. These include hormones, cytokines, chemokines, and growth factors that influence systemic metabolism, cardiovascular function, immune responses, and reproductive function. Key adipokines discussed or implied in the context of EMS include leptin, adiponectin, tumor necrosis factor alpha, TNF, interleukin-6, Illinois-6, and resistin, among others. Alterations in adipokine secretion may contribute to insulin dysregulation by affecting insulin sensitivity, inflammatory tone, energy balance, and appetite regulation. The review suggests that dysregulated adipokine signaling may represent an important link between obesity and insulin dysregulation, although causal relationships remain incompletely defined. The directionality of this relationship is complex. Obesity may cause adipokine dysregulation, which in turn promotes insulin dysregulation, But insulin dysregulation may also promote obesity through its effects on lipogenesis and lipolysis, creating bidirectional causality. Leptin is a hormone produced primarily by adipocytes and plays a central role in regulating energy metabolism, appetite, and energy expenditure. Leptin was originally discovered in mice, where mutations in the leptin gene or leptin receptor gene result in severe obesity and metabolic dysfunction. In normal physiology, leptin acts via regulation of the satiety center in the hypothalamus to inhibit food intake and increase energy expenditure through thermogenesis and increased physical activity. In horses, as in many other species, serum leptin concentrations are positively correlated with adiposity. Multiple studies have demonstrated that obese horses exhibit higher circulating leptin concentrations than horses in moderate body condition. This relationship appears to hold across breeds and age groups, although breed-specific differences in leptin concentrations have been reported. In horses with EMS, elevated leptin levels have been observed, particularly in obese individuals. The review notes that hyperleptinemia, defined as leptin concentrations exceeding 7.3 nanograms per milliliter in some studies, may help predict laminitis risk in ponies. Research by Carter and colleagues found that hyperleptinemia, defined as leptin concentrations exceeding 7.3 nanograms per milliliter, along with hyperinsulinemia, defined as insulin concentrations exceeding 32 microinternational units per liter, could help predict laminitis occurrence. However, the biological interpretation of hyperleptinemia in EMS is complex. Despite elevated leptin concentrations, affected horses often fail to reduce food intake or increase energy expenditure, suggesting a state of leptin resistance. Leptin resistance refers to a condition in which elevated leptin concentrations fail to elicit normal physiological responses. The mechanisms underlying leptin resistance are not fully understood but may involve impaired leptin transport across the blood-brain barrier, downregulation of leptin receptors in the hypothalamus, or post-receptor signaling defects. Furthermore, the level of leptin expression is increased in nuchal ligament adipose tissue, suggesting that local fat accumulation at this site can significantly contribute to elevating the overall level of leptin in horses. The nuchal ligament, which provides structural support to the neck, is infiltrated with adipose tissue in horses with prominent cresty necks. Adipocytes within this depot appear to be particularly active in leptin secretion, and nuchal fat may therefore disproportionately contribute to systemic hyperleptinemia. Adiponectin is another adipokine of interest in EMS research, generally associated with improved insulin sensitivity and anti-inflammatory effects. Unlike leptin, adiponectin concentrations are typically inversely related to adiposity. Lean individuals tend to have higher adiponectin levels than obese individuals. Adiponectin enhances insulin sensitivity in liver and skeletal muscle, promotes fatty acid oxidation, and exerts anti-inflammatory effects on vascular endothelium. In many metabolic disorders, including human metabolic syndrome and type 2 diabetes, adiponectin concentrations are reduced. This hypoadiponectinemia is thought to contribute to insulin resistance and metabolic dysfunction. The review references evidence indicating that horses with EMS may exhibit decreased adiponectin levels, particularly in association with obesity. Reduced adiponectin may exacerbate insulin dysregulation by impairing glucose uptake and lipid metabolism in peripheral tissues. Lower adiponectin concentrations have been associated with reduced insulin sensitivity in horses, although the relationship is not always consistent across studies. This imbalance between adipokines that promote insulin sensitivity, such as adiponectin, and those that contribute to insulin resistance or metabolic dysfunction, such as leptin and pro-inflammatory cytokines, reflects a broader dysregulation of adipose tissue function in EMS. The adipokine profile in EMS is characterized by elevated leptin, reduced adiponectin, and increased pro-inflammatory cytokines, a pattern that promotes metabolic dysfunction. Beyond endocrine signaling, adipose tissue may contribute to systemic metabolic dysfunction through low-grade inflammation and oxidative stress. The review highlights findings suggesting that adipose tissue in EMS-affected horses exhibits increased expression of inflammatory mediators and markers of oxidative stress. Pro-inflammatory cytokines, including TNF, Illinois-6, and monocyte chemoattractant protein 1, MCP1, are secreted by adipocytes and infiltrating immune cells within adipose tissue. These cytokines can interfere with insulin signaling pathways, contributing to insulin resistance. TNF, for example, promotes serine phosphorylation of insulin receptor substrate proteins, which inhibits their normal tyrosine phosphorylation and impairs downstream signaling. Chronic low-grade inflammation may interfere with insulin signaling pathways, further contributing to insulin dysregulation. Inflammatory signaling activates stress kinases such as C-Jun N-terminal kinase, JNK, and inhibitor of B kinase, IKK, which phosphorylate insulin receptor substrate proteins on serine residues, inhibiting their function and impairing insulin signal transduction. Oxidative stress, characterized by an imbalance between reactive oxygen species production and antioxidant defenses, may impair cellular function across multiple tissues. Adipose tissue from obese individuals exhibits increased production of reactive oxygen species, ROS, including superoxide, hydrogen peroxide, and hydroxyl radicals. These reactive molecules can damage cellular lipids, proteins, and DNA, impairing normal cellular function. While the degree of inflammation observed in EMS is generally mild compared to overt inflammatory diseases, its chronic nature may have cumulative effects on metabolic health. The low-grade, persistent inflammatory state associated with obesity and insulin dysregulation has been termed meta-inflammation in human medicine, reflecting its metabolic origins and consequences. Bruinstein and colleagues demonstrated that expression of inflammation-related genes is associated with adipose tissue location in horses. Nuchal fat exhibited higher expression of inflammatory markers compared to subcutaneous fat from other locations, suggesting site-specific differences in inflammatory activity. This finding provides additional support for the concept that regional adiposity, particularly nuchal fat accumulation, may be particularly problematic from a metabolic standpoint. We shall now look at adipose-derived stem cells, cellular dysfunction in EMS. One of the more specialized topics addressed in the review concerns adipose-derived stem cells, abbreviated ASCs. These multipotent cells reside within adipose tissue and contribute to tissue maintenance, repair, and regeneration. ASCs can differentiate into multiple cell types, including adipocytes, osteoblasts, chondrocytes, and myocytes, under appropriate conditions. Studies cited in the review indicate that ASCs from EMS-affected horses exhibit altered proliferative capacity, increased oxidative stress, and signs of cellular senescence. Research by Marich and colleagues has extensively characterized the dysfunction of ASCs in EMS. These investigations have revealed multiple abnormalities, including increased surface marker expression of CD44+, which stimulates inflammatory reactions in adipose tissue, higher numbers of mitochondrial abnormalities, including disrupted cristae structure and swollen mitochondria, elevated nitric oxide (NO) production, higher reactive oxygen species (ROS) production and accumulation, reduced superoxide dismutase (SOD) activity, indicating impaired antioxidant defense, deregulated expression of molecular markers related to apoptosis, autophagy, regenerative ability, elimination of dysfunctional mitochondria, and cellular metabolism. Specific molecular markers found to be dysregulated in ASCs from EMS horses include P21, a cell cycle inhibitor associated with senescence, BCL2-BAX ratio, which regulates apoptosis, BMP2, involved in osteogenic differentiation, PGC1-alpha, a master regulator of mitochondrial biogenesis, PARC1, involved in mitochondrial quality control, and PDK4, which regulates glucose oxidation. These changes collectively indicate profound mitochondrial dysfunction and cellular stress. Because of oxidative stress, apoptosis, and mitochondrial function deterioration, ASCs from EMS-affected horses possess a senescent phenotype with enlarged cell bodies and nuclei, increased apoptosis rates, and reduced heterochromatin architecture. Cellular senescence refers to a state of permanent cell cycle arrest accompanied by altered gene expression and secretion of inflammatory mediators. Senescent cells accumulate with age and in response to various stresses, and their accumulation contributes to tissue dysfunction. However, the review notes that ASCs from EMS horses develop enhanced autophagy during early stages of differentiation to maintain cellular homeostasis, allowing for removal of ROS-induced damage. Autophagy is a cellular process involving degradation and recycling of damaged organelles and proteins. This compensatory increase in autophagy may represent an adaptive response to metabolic stress, attempting to mitigate oxidative damage and maintain cellular function. Although the clinical implications of ASC dysfunction are not yet fully understood, these findings provide insight into the broader systemic effects of metabolic dysregulation. They suggest that EMS affects not only metabolic function but also tissue regenerative capacity, potentially compromising the horse's ability to repair damaged tissues. This has implications for recovery from injury, wound healing, and long-term tissue health. The potential therapeutic application of ASCs in EMS has also been explored. Some research has investigated whether transplantation of modified or rejuvenated autologous ASCs could improve metabolic function or reduce tissue damage in EMS-affected horses. While this represents an area of active investigation in regenerative medicine, clinical applications remain experimental. Further investigation of other tissues in EMS-affected horses has revealed that adipose and liver tissues are particularly affected by stress induced via p53 signaling, endoplasmic reticulum stress, and autophagy, which in turn protects cells against glucolipotoxicity. Glucolipotoxicity refers to cellular dysfunction caused by simultaneous exposure to elevated glucose and lipid concentrations. The endoplasmic reticulum is a cellular organelle responsible for protein folding and lipid synthesis. When the demand for protein folding exceeds capacity, unfolded or misfolded proteins accumulate, triggering endoplasmic reticulum stress. Chronic endoplasmic reticulum stress activates signaling pathways that can lead to inflammation, insulin resistance, and apoptosis. Interestingly, in EMS muscles, other mechanisms must occur to protect cells from insulin dysfunction-related apoptosis. Skeletal muscle from EMS horses does not show the same degree of endoplasmic reticulum stress or autophagy activation observed in adipose and liver tissues. This tissue-specific response suggests that different organs employ different adaptive strategies to cope with metabolic stress. The liver plays a central role in glucose homeostasis, lipid metabolism, and insulin clearance, and hepatic dysfunction may contribute to EMS pathophysiology. Some studies have suggested that in obese and insulin-resistant horses, the liver may play an important role in disease progression. The liver impairment in EMS horses at the molecular level is manifested by increased apoptosis, increased endoplasmic reticulum stress, oxidative stress, excessive accumulation of lipids, hepatic steatosis, and increased inflammation. Hepatic steatosis, or fatty liver, occurs when triglyceride accumulation exceeds the liver's capacity for fatty acid oxidation and very low-density lipoprotein secretion. Several studies in the field of regenerative veterinary medicine have reported the possibility of using transplantation of modified autologous adipose stem cells to reduce the impairment of the liver. This experimental approach attempts to leverage the regenerative and immunomodulatory properties of ASCs to improve hepatic function. While promising in experimental studies, clinical application remains in early stages of development. An important nuance emphasized by the authors is that obesity and EMS are related but distinct conditions. Not all obese horses develop insulin dysregulation, and not all horses with EMS are obese. The 2019 ASEAN Consensus Statement explicitly notes that obesity, while frequently associated with EMS, is very misleading as a diagnostic criterion and should not be considered a required feature for diagnosis. This distinction highlights the importance of individual susceptibility and genetic background. Some horses may tolerate increased adiposity without developing metabolic dysfunction, whereas others may develop EMS at relatively modest levels of fat accumulation. This variability complicates efforts to define clear diagnostic thresholds based on body condition alone and reinforces the need for metabolic testing rather than reliance on phenotypic assessment. The relationship between obesity and insulin dysregulation in EMS is bidirectional. Obesity may promote insulin dysregulation through adipokine imbalance, inflammation, oxidative stress, and lipotoxicity, while hyperinsulinemia promotes fat accumulation by stimulating lipogenesis and inhibiting lipolysis. This self-reinforcing cycle contributes to the persistence and progression of EMS once established. Breaking this cycle through dietary management, caloric restriction, and increased physical activity is a central goal of EMS management, although detailed discussion of therapeutic interventions falls outside the primary focus of the genetic review. In summary, obesity in equine metabolic syndrome is best understood not simply as excess body weight, but as a complex alteration of adipose tissue biology. Regional fat accumulation, particularly in the nuchal region, dysregulated adipokine secretion including hyperleptinemia and hypoadiponectinemia, low-grade chronic inflammation, oxidative stress, and stem cell dysfunction collectively contribute to the metabolic phenotype observed in EMS. The review by Stefaniuk-Szmukier et al. Positions obesity as an important modifier of disease expression rather than a universal or defining feature. This nuanced perspective aligns with the broader theme of EMS as a multifactorial and heterogeneous syndrome. Understanding the active role of adipose tissue in disease pathogenesis, rather than viewing it as passive energy storage, is essential for developing effective prevention and treatment strategies. We now move on to genetic factors and the molecular basis of equine metabolic syndrome. The investigation of genetic factors underlying equine metabolic syndrome represents a relatively recent but rapidly expanding area of research. In the review by Stefaniuk-Szmukier, Piórkowska, and Ropka-Molik, 2023, genetic predisposition is presented not as a singular causal determinant, but as a foundational influence that modulates an individual horse's susceptibility to metabolic dysfunction in response to environmental and management-related factors. This section examines the genetic architecture of EMS in detail, emphasizing the multifactorial and polygenic nature of the syndrome. The authors are careful to avoid simplistic genetic explanations, instead framing EMS as the outcome of numerous interacting genetic variants, each contributing modest effects to the overall phenotype. The existence of a genetic component to EMS is supported by several lines of evidence. One of the most frequently cited observations is the uneven distribution of EMS prevalence across breeds. Certain breeds, particularly ponies and native or primitive horse breeds, are disproportionately affected by EMS and its associated traits. Breeds commonly identified as having increased susceptibility include Welsh ponies of all sections, Shetland ponies, Morgan horses, Icelandic horses, Norwegian fjord horses, Spanish mustangs, Paso-Finos, and various other native breeds adapted to harsh environments with limited nutritional availability. Breed surveys and clinical studies have consistently demonstrated higher prevalence of obesity, insulin dysregulation, and laminitis in these populations compared to other breed types. In contrast, some sport horse breeds, including thoroughbreds, warm bloods, and standardbreds, appear to exhibit lower prevalence of EMS, although this observation may be confounded by differences in management practices and selection pressures. Sport horses are typically maintained under management systems emphasizing athletic conditioning, regular exercise, and controlled feeding, whereas ponies are often kept as companion animals with less structured exercise and ad libitum access to pasture. The review emphasizes that breed predisposition does not equate to determinism. Rather, it reflects population-level differences in allele frequencies for genes involved in energy metabolism, insulin signaling, adipose tissue function, and related pathways. Individual animals within high-risk breeds may not develop EMS, while animals from lower-risk breeds occasionally do, reflecting the complex interplay of genetic and environmental factors. Heritability estimates provide quantitative insight into the genetic contribution to EMS-related traits. Heritability, in the narrow sense, refers to the proportion of phenotypic variance in a population that can be attributed to additive genetic effects. Values range from 0, indicating no genetic contribution, to 1, indicating that all variation is genetic. The authors summarize studies indicating moderate heritability for phenotypes such as insulin concentration, glucose tolerance, body condition score, and regional adiposity. These estimates typically range from 0.2 to 0.5, depending on the specific trait, population studied, and methodology employed. For example, Norton and colleagues estimated SNP-based heritability for various metabolic traits in Morgan horses and Welsh ponies. In Welsh ponies, 7 biochemical parameters related to EMS diagnosis showed significant heritability estimates, with ACTH concentration showing the highest heritability. In Morgan horses, 6 of the analyzed traits, excluding glucose concentrations, triglycerides, and ACTH, had significant heritability estimates. These findings suggest that genetic variation accounts for a meaningful proportion of phenotypic variability, although environmental factors remain critically important. Importantly, heritability estimates vary depending on the trait measured, the population studied, and the testing methodology employed. Pedigree analysis has also provided evidence for genetic contributions to EMS. Treiber and colleagues evaluated pasture-associated laminitis in a herd of ponies with detailed pedigree records. The pattern of laminitis occurrence among related animals suggested a dominant pattern of inheritance of a major gene or genes, although with reduced penetrance. Reduced penetrance means that not all animals carrying risk alleles develop disease, likely due to the influence of environmental factors, sex, age of onset, and possible epigenetic modifications. This variability underscores the complexity of EMS as a phenotype and cautions against overinterpretation of individual genetic markers. The presence of risk alleles increases susceptibility but does not guarantee disease development, while their absence does not confer absolute protection. One of the central conclusions of the review is that EMS is a polygenic condition, meaning it arises from the combined effects of many genes rather than from mutations in a single gene of large effect. Each contributing genetic variant may exert only a small influence on metabolic function, but collectively these variants shape an individual's metabolic efficiency, insulin responsiveness, adipogenic capacity, and inflammatory tone. McHugh and colleagues proposed a genetic model underlying EMS that has been influential in the field. According to this model, the phenotypic components of EMS involve mechanisms regulating insulin, glucose, and lipid metabolism that are part of complex metabolic pathways with multiple and numerous alleles. These alleles, in turn, are influenced by epigenetic modification under environmental pressure. Following this framework, the genetic background responsible for the risk of EMS is a unique combination of several loci. This assumption can also be used to understand breed differences in susceptibility to EMS. Different breeds may share some common risk alleles but differ in others, and the specific combination of alleles present in an individual determines overall risk. This polygenic architecture complicates both research and clinical application, as it precludes straightforward genetic testing for EMS based on a single mutation or gene. Unlike monogenic diseases such as hyperkalemic periodic paralysis or severe combined immunodeficiency, for which single gene tests have been developed, EMS requires assessment of multiple genetic variants, each with small individual effects, to predict risk accurately. Early genetic studies of EMS often employed candidate gene approaches, focusing on genes known or suspected to be involved in metabolic regulation. These studies examined polymorphisms in genes related to insulin signaling, lipid metabolism, inflammation, and energy balance. Candidate genes investigated in equine metabolic research have included genes encoding components of the insulin signaling pathway, insulin receptor substrate proteins, glucose transporters. Genes involved in lipid metabolism, lipoprotein lipase, fatty acid binding proteins, perilipin. Genes encoding adipokines, leptin, adiponectin. Genes involved in inflammatory signaling, tumor necrosis factor alpha, interleukins. While some associations were identified through candidate gene studies, the review notes that this approach is inherently limited by prior assumptions about gene function and may overlook novel or unexpected contributors. Candidate gene studies typically have limited power to detect variants of small effect and may produce false positive associations due to population stratification or multiple testing. As a result, the field has increasingly shifted toward genome-wide approaches that do not require prior hypotheses about specific genes. Genome-wide association studies, abbreviated GWAS, represent a major methodological advance in the study of EMS genetics. GWAS analyze hundreds of thousands to millions of genetic markers, typically single nucleotide polymorphisms, SNPs, distributed across the entire genome to identify associations with specific phenotypes. The review summarizes several GWAS that have identified genomic regions associated with EMS-related traits, including insulin dysregulation and laminitis risk. Notably, these studies often identify loci, meaning chromosomal regions, rather than specific causal genes. The SNPs included on genotyping arrays are typically not themselves causative, but rather serve as markers in linkage disequilibrium with nearby causal variants. One of the key loci discussed in detail involves the gene FAM174A, also known as NS5-ATP6 or TMEM157. This gene has been associated with insulin concentration and laminitis susceptibility in several populations, particularly Arabian horses. We need to discuss the FAM174A gene and associated variants. Lewis and colleagues conducted a GWAS in Arabian horses that revealed three promising genetic markers: BAEC2-263370, BAEC2-263373, and BAEC2-263524. These SNPs are located within the FAM174A gene region on equine chromosome 6. The study found that these markers were potentially associated with elevated insulin levels, insulin resistance, and laminitis occurrence, as well as body condition evaluation indices. The biological function of FAM174A is not fully understood, but research suggests it may play a role in lipid regulation. Studies in human cell lines have shown that knockdown of FAM174A results in downregulation of low-density lipoproteins and increases in cellular free cholesterol. Conversely, overexpression of FAM174A affects triglyceride content. Perhaps most importantly, FAM174A appears to interact with fibroblast growth factor 21, abbreviated FGF21. FGF21 is an important metabolic regulator that enhances insulin sensitivity, acts as an activator of glucose uptake in adipocytes, protects against diet-induced obesity, and serves as a crucial mediator of the adaptive starvation response. Research has demonstrated that FAM174A transcriptionally regulates FGF21 through promoter activation, and also regulates FGF21 expression post-transcriptionally through upregulation of microRNA 577. The interaction between FAM174A and FGF21 provides a plausible mechanistic link to EMS pathophysiology. Variants in FAM174A that alter its expression or function could theoretically affect FGF21 levels, which in turn would influence insulin sensitivity, adipose tissue function, and metabolic efficiency. A subsequent study by Stefaniuk-Szmukier and colleagues in a Polish population of Arabian horses found a high frequency, 51%, of the BIEC2-263524 risk allele, which correlated with higher insulin levels and laminitis occurrence. This frequency is substantially higher than that observed in some other breeds, consistent with the increased EMS prevalence in Arabians. However, breed-specific studies have yielded variable results. Cash and colleagues investigated Australian ponies with known metabolic status and questioned the strong association of FAM174A with EMS in this breed. The frequency of the risk allele was low, and among affected ponies, only 15% possessed the FAM174A risk variant. Most affected ponies showed obesity, but there was no association between obesity and FAM174A genotypes. This result differed from that presented in Arabian horses, where obesity was proposed as one of the main phenotypic features related to the FAM174A risk allele. The discrepancy highlights important considerations. Genetic associations may be breed-specific due to differences in linkage disequilibrium patterns, genetic background, and allele frequencies. A variant that is strongly associated with EMS in one breed may be weakly associated or not associated in another breed, even if the underlying causal variant is the same, due to differences in linkage relationships. We shall now look at additional genomic regions and candidate genes. Beyond FAM174A, GWAS in Morgan horses and Welsh ponies have identified additional genomic regions of interest. Schultz and colleagues, in work described in Norton et al. 2019, applied GWAS analyses using the Illumina SNP50 chip to a cohort of 286 Morgan horses and 264 Welsh ponies. This work allowed for narrowing down the search area to 17 genome-wide significant candidate loci for 9 metabolic traits and 2 morphometric measurements. The top-ranked quantitative trait locus, QTL, for the investigated breeds was located within 1 megabase of the ZFAT and HMGA2 genes. Genes overlapping with human metabolic syndrome regions included VEGFA, NRXN3, GRIK2, TRIB2, AHR, and ISL1, all of which are potentially related to EMS risk. Notably, the AHR gene, which encodes the aryl hydrocarbon receptor, was associated with regulation of energy metabolism and metabolic syndrome in humans. The aryl hydrocarbon receptor is a transcription factor that responds to environmental toxins, but also plays roles in cellular differentiation and metabolic regulation. Genome-wide significant loci for fasting glucose were found near the CRYBA4 and DLGAP4 genes, while those for fasting insulin levels were near GRIK2 and ATG14. GWAS results for glucose levels 75 minutes post-oral sugar test indicated the SPAG16 and HDAC1 genes, while insulin levels 75 minutes post-oral sugar test indicated the ISL1 and RNF217 genes. The identification of multiple loci across the genome reinforces the polygenic nature of EMS. No single genomic region accounts for a large proportion of variance in EMS-related traits. Instead, risk is distributed across many loci, each contributing modestly to overall susceptibility. The HMGA2 gene, which encodes high-mobility group AT-HOOK2 protein, has received particular attention for its pleiotropic effects on both height and metabolic traits. HMGA2 contains 3 AT-hook DNA binding motifs, acts as a transcription regulator, and is involved in adipogenesis and diet-induced obesity. Norton and colleagues evaluated a specific HMGA2 variant, HMGA2 C.83G greater than A, PG28E, for pleiotropic effects on height and metabolic traits in ponies. This variant was associated with reduced height and also showed associations with insulin dynamics. The study demonstrated that baseline insulin levels are inversely correlated with height in ponies, a relationship that has been well described in humans, where shorter stature is associated with increased risk of type 2 diabetes. Correlation analyses between height and several biochemical parameters in ponies indicated a significant inverse correlation between insulin, glucose, adiponectin, and ACTH levels, and a positive correlation between height and triglycerides and leptin concentrations. In a GWAS, 2 loci on equine chromosome 6 contributed roughly 40% and 20% of variance in height and insulin levels, respectively. The HMGA2 C.83G greater than a variant was previously shown to reduce overall size in Shetland ponies. The finding that this same variant influences insulin dynamics suggests true pleiotropy, wherein a single genetic variant affects multiple seemingly unrelated traits. This relationship has important implications for breeding. Selection for small size may inadvertently increase metabolic risk. The estimation of SNP-based heritability across multiple traits provides insight into the genetic architecture of EMS. Studies conducted on Morgan horses and Welsh ponies estimated heritability for 9 traits measured according to EMS diagnosis: glucose, glucose OST, oral sugar test, insulin, insulin OST, non-esterified fatty acids, NEFA, serum concentrations of triglycerides, adiponectin, leptin, and ACTH. In Welsh ponies, 7 biochemical parameters reached significant heritability estimates, with ACTH showing the highest heritability. In Morgan horses, 6 of the analyzed traits had significant heritability estimates, excluding glucose concentrations, triglycerides, and ACTH. This finding provides strong evidence of genetic contribution to EMS-related traits while also indicating differences among breeds in the genetic architecture of specific traits. The differences in heritability patterns between breeds suggest that the specific genes and pathways contributing to EMS risk may vary across populations. This genetic heterogeneity complicates efforts to develop universal genetic tests, but also provides opportunities for breed-specific risk assessment. A recurring theme in the review is the importance of gene-environment interactions. Genetic predisposition alone is insufficient to cause EMS. Rather, genetic susceptibility interacts with environmental exposures such as diet composition, caloric intake, physical activity, and seasonal variation to determine disease risk. Horses with a genetic predisposition for efficient energy storage and heightened insulin responsiveness may remain metabolically healthy under conditions of limited nutrition and regular exercise, but develop EMS when exposed to energy-dense diets and minimal activity. This interaction-based model aligns with a broader understanding of complex metabolic diseases across species and reinforces the need for integrative approaches to prevention and management. The concept of gene-environment interaction has important practical implications. It suggests that EMS is not inevitable, even in genetically predisposed animals, if appropriate environmental management is maintained. Conversely, even animals with lower genetic risk may develop EMS if environmental factors are sufficiently adverse. The genetic variants associated with EMS-related traits implicate several molecular pathways, including insulin signaling and glucose transport, lipid metabolism and adipogenesis, inflammatory and oxidative stress pathways, cellular growth, proliferation and differentiation, transcriptional regulation and chromatin remodeling. Rather than pointing to a single disrupted pathway, the genetic data suggest widespread modulation of metabolic networks, consistent with the multifactorial nature of the syndrome. Integration of genetic data with transcriptomic and metabolomic data may help clarify how specific genetic variants influence molecular pathways and cellular function. The review is careful to acknowledge the limitations of existing genetic studies. These include relatively small sample sizes, which limit statistical power to detect variants of small effect, limited breed representation, with most studies focusing on a small number of breeds, challenges in phenotypic classification, as EMS diagnosis involves multiple criteria and dynamic testing, and the fact that many identified loci explain only a small proportion of phenotypic variants, limiting their predictive utility at the individual level. Additionally, most GWAS identify associations at the population level but do not prove causality. The identified SNPs are typically markers in linkage disequilibrium with causal variants rather than being causal themselves. Fine mapping and functional studies are required to identify the actual causal variants and understand their biological effects. The authors emphasize that genetic findings should be interpreted as risk modifiers rather than deterministic predictors. The presence of risk alleles increases susceptibility but does not guarantee disease development, while their absence does not confer absolute protection. Looking forward, the review advocates for larger, multi-breed studies that can provide adequate statistical power to detect variants of small effect and allow for cross-breed validation of findings. Integration of genomic data with transcriptomic and metabolomic analyses will enable more comprehensive understanding of molecular mechanisms. Improved phenotyping of EMS traits, including standardized diagnostic criteria and dynamic testing protocols, will enhance the quality of genetic studies. Development of polygenic risk scores that integrate information from multiple genetic variants may eventually enable personalized risk assessment. Functional studies to validate candidate genes and elucidate mechanisms will be essential for translating genetic discoveries into practical applications. Such integrative approaches may eventually clarify the molecular mechanisms underlying genetic associations and enable more precise risk stratification. However, the review acknowledges that significant challenges remain, and practical application of genetic testing for EMS risk assessment is not yet routine in clinical practice. Let's transition to diagnostic implications, knowledge gaps, and future research directions. The final section of the review by Stefaniuk-Szmukier, Piórkowska, and Ropka-Molik, 2023, synthesizes the preceding discussions into a broader consideration of how current knowledge of equine metabolic syndrome informs diagnosis, highlights existing gaps in understanding, and guides future research priorities. This section does not introduce fundamentally new concepts, but rather revisits and reiterates key themes in a more integrative and forward-looking context. One of the recurring issues emphasized throughout the review is the inherent difficulty of diagnosing EMS with precision and consistency. The syndrome lacks a single definitive diagnostic test, relying instead on the identification of characteristic metabolic features, most notably insulin dysregulation, in conjunction with clinical context and, sometimes, phenotypic features. The variability in clinical presentation complicates diagnostic efforts. Some horses exhibit overt obesity and regional adiposity, presenting a clear clinical picture, while others appear phenotypically normal despite significant underlying metabolic dysfunction. Similarly, insulin dysregulation may be evident under certain testing conditions but not others, depending on dietary status, time of year, time of day, and the specific test employed. As a result, diagnosis often depends on a combination of clinical assessment, laboratory testing, risk evaluation based on breed and management, and clinical judgment, rather than on a singular diagnostic threshold. This inherent ambiguity can lead to underdiagnosis in subclinical cases and may delay intervention until after laminitis has developed. The review underscores that the assessment of insulin dysregulation is central to the diagnosis of EMS. Various testing approaches are used in clinical and research settings, each with distinct advantages and limitations. Fasting insulin measurement represents the simplest and most practical approach. A blood sample is collected after a period of fasting, typically 6 to 12 hours, and insulin concentration is measured. However, fasting insulin concentrations may be normal in some EMS-affected horses, particularly in early stages of disease or in animals with primarily gastrointestinal-driven insulin dysregulation. The ECEIM consensus statement suggests a cutoff value of greater than 20 microinternational units per milliliter as indicative of fasting hyperinsulinemia, although specific thresholds may vary depending on assay methodology. Oral sugar tests have become widely adopted in clinical practice due to their practicality and physiological relevance. As described previously, these tests involve oral administration of corn syrup or dextrose solution, followed by serial insulin measurements at 60 and 90 minutes. The test mimics natural feeding conditions and stimulates incretin release, potentially unmasking insulin dysregulation that is not apparent under fasting conditions. The ECEIM consensus statement suggests that insulin concentrations exceeding 60 microinternational units per milliliter at 60 or 90 minutes indicate insulin dysregulation. Intravenous glucose tolerance tests provide more detailed information about insulin dynamics but are more complex and labor-intensive to perform. These tests involve intravenous administration of glucose followed by frequent blood sampling to assess glucose and insulin responses over time. The frequently sampled intravenous glucose tolerance test, combined with minimal model analysis, allows calculation of insulin sensitivity and glucose effectiveness. However, these tests require multiple blood samples, mathematical modeling, and specialized expertise. Combined glucose-insulin tests involve administration of both glucose and insulin, typically by intravenous injection, followed by serial blood glucose measurements. These tests assess tissue responsiveness to insulin, but do not directly measure insulin secretion. Each method has advantages and limitations. Fasting insulin measurements are simple but may lack sensitivity. Oral testing methods may reveal exaggerated insulin responses that are not apparent under fasting conditions but are influenced by gastrointestinal function and incretin responses. Intravenous tests provide detailed information about insulin dynamics but bypass the gastrointestinal tract and may not reflect real-world feeding conditions. The authors emphasize that insulin dysregulation should be viewed as a continuum rather than a binary state, further complicating the establishment of universal diagnostic cutoffs. Some horses may have borderline results that do not clearly fall into affected or unaffected categories. Repeat testing, consideration of seasonal influences, and clinical judgment are often necessary. Phenotypic evaluation remains an important component of EMS diagnosis, particularly in identifying horses at increased risk. Body condition scoring using the Hennig system and assessment of regional adiposity using Cresty neck scoring provide practical tools for identifying animals that may warrant further metabolic testing. However, as noted throughout the review, phenotypic features alone are insufficient for diagnosis. The absence of obesity does not exclude EMS, as some horses develop insulin dysregulation without marked weight gain, particularly those with predominantly gastrointestinal mechanisms of hyperinsulinemia. Conversely, obesity alone does not confirm EMS, as some obese horses maintain normal insulin dynamics. This nuanced relationship reinforces the need for integrated diagnostic approaches that combine phenotypic assessment with metabolic testing. Visual evaluation and body condition scoring serve as screening tools to identify at-risk individuals who should undergo more definitive testing, but they cannot replace laboratory assessment of insulin dynamics. One of the key questions addressed in the review is whether genetic information can enhance diagnostic accuracy for EMS. While genetic predisposition is clearly an important component of disease risk, the authors caution that current genetic knowledge is not yet sufficient for routine clinical testing. The polygenic nature of EMS, combined with breed-specific genetic architecture, limits the predictive value of individual genetic markers. A single SNP genotype cannot reliably predict whether an individual horse will develop EMS because risk depends on the combined effects of many genetic variants, each contributing small effects in interaction with environmental factors. At present, genetic findings are more useful for understanding disease mechanisms and population-level risk than for individual diagnosis. Knowledge of breed predisposition can inform clinical suspicion and guide testing recommendations, but individual genetic testing is not currently recommended as a diagnostic tool. Nevertheless, the authors suggest that future advances in genomic technologies may eventually allow for more refined risk stratification. Development of polygenic risk scores that integrate information from multiple genetic variants, validated across diverse populations, could potentially enable personalized assessment of EMS risk. Such tools would need to account for breed-specific genetic architecture and gene-environment interactions to be clinically useful. Although the review focuses primarily on genetic and pathophysiological aspects of EMS, it implicitly highlights the importance of early identification in preventing clinical consequences such as laminitis. Recognizing metabolic risk before the onset of laminitis allows for the implementation of management strategies aimed at controlling insulin dysregulation. While specific management interventions fall outside the scope of the genetic review, the underlying principle of prevention is repeatedly emphasized. Proactive screening of at-risk populations, particularly ponies and native breeds, individuals with regional adiposity, animals with a history of laminitis, and horses maintained on high-carbohydrate diets, can identify metabolic dysfunction before irreversible complications develop. This preventive perspective aligns with the broader goal of improving equine welfare through proactive metabolic monitoring. Regular metabolic assessment, particularly during high-risk seasons such as spring and early summer, can detect worsening insulin dynamics and prompt intervention before laminitis occurs. The authors explicitly identify several areas where knowledge remains incomplete. These include the precise molecular mechanisms by which hyperinsulinemia induces laminitis, including the relative contributions of IGF-1 receptor activation, vascular dysregulation, altered cellular metabolism, advanced glycation end products, and matrix metalloproteinase activation. The relative contributions of insulin resistance versus insulin hypersecretion versus altered insulin clearance to hyperinsulinemia in EMS. The functional roles of specific genes identified through GWAS, including how genetic variants influence gene expression, protein function, and metabolic phenotypes. The interactions between genetic predisposition and environmental exposure, including gene-environment interactions, gene-diet interactions, and epigenetic modifications. The mechanisms underlying breed differences in EMS susceptibility. The role of the microbiome in insulin dysregulation and incretin responses. The temporal progression from normal metabolism to insulin dysregulation to clinical laminitis. The factors that determine individual variation in laminitis susceptibility among horses with similar insulin dysregulation. These gaps reflect both the complexity of EMS and the limitations of current research methodologies. Addressing these questions will require diverse approaches, including in vitro cellular studies, in vivo animal experiments, large-scale epidemiological studies, and translational research integrating findings across different levels of biological organization. In addition to conceptual gaps, the review acknowledges methodological challenges in EMS research. These challenges include small sample sizes in genetic studies, which limit statistical power to detect variants of small effect and increase the risk of false positive findings. Heterogeneous populations that may include multiple breeds or mixed breed animals, complicating genetic analysis and interpretation. Variable phenotyping across studies, with differences in diagnostic criteria, testing protocols, and thresholds for defining affected status. Lack of standardized definitions and testing protocols, which complicate comparisons and synthesis of results across studies. Limited longitudinal data tracking metabolic changes over time, making it difficult to understand disease progression and temporal relationships. The authors suggest that standardized definitions and testing protocols would enhance the robustness of future research. Development of consensus diagnostic criteria, similar to the ASEAN statement but with additional specificity, would facilitate more consistent phenotyping across studies and enable meaningful comparisons of results. Looking forward, the review advocates for integrative research approaches that combine genomic, transcriptomic, and metabolomic data. Multi-omic strategies may provide a more comprehensive understanding of the molecular networks underlying EMS. Genomics can identify genetic variants associated with EMS risk. Transcriptomics can reveal how these variants influence gene expression patterns in relevant tissues such as adipose tissue, liver, and muscle. Metabolomics can characterize the downstream metabolic consequences of genetic and transcriptomic changes. Integration of these data layers can help connect genetic variants to molecular mechanisms to phenotypic outcomes. The authors also emphasize the importance of longitudinal studies that track metabolic changes over time. Most existing studies employ cross-sectional designs, comparing affected and unaffected animals at a single time point. Longitudinal studies that follow animals from healthy status through development of insulin dysregulation and, in some cases, laminitis would provide insight into disease progression, identify early biomarkers, and reveal temporal relationships between different components of the syndrome. Collaborative efforts involving multiple breeds and populations are also essential. Genetic architecture may vary across breeds, and findings from single breed studies may not generalize to other populations. Large-scale, multi-breed consortia similar to those employed in human genome-wide association studies could provide the sample sizes and diversity needed to identify shared and breed-specific genetic risk factors. These approaches are expected to refine understanding of disease progression and improve identification of at-risk individuals. Ultimately, Such research may enable personalized approaches to EMS prevention and management, accounting for individual genetic risk, environmental exposures, and specific metabolic phenotypes. In synthesizing current knowledge, the review reinforces the conceptualization of EMS as a complex, multifactorial syndrome rather than a single disease entity. Genetic predisposition, insulin dysregulation, adipose tissue biology, incretin signaling, hepatic function, seasonal influences, dietary composition, physical activity, and management practices interact dynamically to shape disease expression. This integrative view challenges simplistic diagnostic models based on single criteria and highlights the need for individualized assessment and management. No two horses with EMS are exactly alike. They may differ in the relative contributions of insulin resistance versus hypersecretion, the degree of obesity and regional adiposity, the presence and severity of laminitis, the specific genetic risk variants they carry, and their responses to dietary and management interventions. Understanding EMS as a systems-level disorder requires appreciation of these multiple interacting components and recognition that intervention at any level, genetic, metabolic, dietary, or management, can influence outcomes. This complexity is daunting but also provides multiple potential targets for prevention and therapy. In conclusion, the review by Stefaniuk-Szmukier et al. Presents EMS as a paradigmatic example of a complex metabolic syndrome shaped by both genetic and environmental influences. While substantial progress has been made in elucidating its components, significant challenges remain in translating this knowledge into precise diagnostic and preventive tools. The authors conclude with a cautious optimism that continued advances in genetic and molecular research will ultimately enhance understanding and management of EMS. The identification of genetic risk factors, elucidation of molecular mechanisms, development of sensitive diagnostic tests, and implementation of evidence-based management strategies all contribute to improving outcomes for affected horses. However, the road ahead is long. The polygenic nature of EMS, breed-specific genetic architecture, gene-environment interactions, and individual variation in disease expression all present formidable challenges. Addressing these challenges will require sustained research effort, interdisciplinary collaboration, large-scale studies, and integration of findings across multiple levels of biological organization. Nevertheless, each advance in understanding brings us closer to effective prevention and management. As knowledge accumulates, clinicians will be better equipped to identify at-risk individuals, implement targeted interventions, and prevent the devastating consequences of EMS-associated laminitis. Thank you for your attention during this session of the Boring Lecture Series. This has been Lecture 5, focused on equine metabolic syndrome as a complex disease influenced by multifactorial genetic factors. The lecture was based on the paper by Stefaniuk-Szmukier, Piórkowska, and Ropka-Molik, published in Genes, Basel, Volume 14, Issue 8. This lecture was narrated by Dr. Owen On, an artificial voice for the Boring Lecture Series. Please see the description for further resources and links to the primary literature cited throughout this discussion. If you found this level of detail sufficient, please consider subscribing for our weekly sessions. We will now fade to soothing bioacoustic sounds and drones to allow for a period of quiet reflection or continued rest. Good night.